Dear students, very good morning to all. Hope you all are fine. Today's our offline class is about mutagens. What are mutagens? Mutations can be artificially produced by certain agents called mutagens or mutagenic agents. Following are two major types of mutagens. Number one, physical mutagens, and number two, chemical mutagens. Now, physical mutagens. Radiations are the most important physical mutagens. A.J. Muller, who used X-rays for the first time to increase the rate of mutation in Drosophila, opened an entirely new field in inducing mutations. So Muller is considered as father of actinobiology. The main sources of spontaneous mutations are the natural radiations coming from cosmic rays of the sun. The spectrum of wavelengths that are shorter, that is, of higher energy than the visible light, can be subdivided into following two groups. Number A, ionizing radiations. Number B, non-ionizing radiations. They occur in small amounts in the environment and are known as background radiations. The following are biological effects of radiations. Number A, effects of ionizing radiations. The ionizing radiations include X-rays, gamma rays, alpha rays, and beta rays. Ionizing radiations cause breaks in the chromosomes. These cells then show abnormal cell divisions. If these include gametes, they may be abnormal or even die prematurely. Different types of cancers may result due to radiations. The frequency of induced mutations is directly proportional to the doses of radiations. Now, B, effects of non-ionizing radiations. The non-ionizing radiations have longer wavelengths but carry lower energy. This energy is insufficient to induce ionization. Therefore, non-ionizing radiations such as UV light do not penetrate beyond the human skin. Thymine pyrimidine dimer formation is a major mutagenic effect of UV rays that disturbs DNA double helix and thus DNA replication. Second type of mutagens, chemical mutagens. Large number of chemical mutagens are now known. These are more injurious than radiations. The first chemical mutagen used was mustard gas by C. Auerbach et al. During World War II. Chemical mutagens are placed into two groups. Number A, those which are mutagenic to both replicating and non-replicating DNA such as nitrous acid, and number B, those which are mutagenic only to replicating DNA such as acridine dyes and base analogues. Following are the effects of some of the chemical mutagens. Number 1, nitrous acid. It is mutagenic to both replicating and non-replicating DNA. It acts directly by oxidative deamination on A, G, and C bases which contain amino groups. A is deaminated to hypoxanthine, which is complementary to cytosine. G is converted to xanthine, which pairs with C. Cytosine is converted to U, which pairs with A. Number 2, acridines. Acridines and proflavins are very powerful mutagens. These can intercalate between DNA bases and interfere with DNA replication, producing insertion or deletion or both of single bases respectively. This includes frame shift mutations or gibberish mutations, for example, thalassemia. Base analogues. These have structures similar to the normal bases and are incorporated into DNA only during DNA replication. Base analogues cause mispairing and eventually give rise to mutations. The base analogues may either be natural or artificial. Natural base analogues include 5-methyl cytosine, 5-hydroxymethyl cytosine, 6-methylpurine, etc. The most commonly used artificial base analogues are 5-bromouracil and 2-aminopurine. 5-bromouracil is a structural analog of thymine. It gets incorporated into the newly replicated DNA in place of thymine T. 2-aminopurine is an artificial base analog of adenine. It acts as a substitute of adenine A and can also pair with cytosine C. This was all about today's after class. Thanks.